Great. Well, welcome everyone. Greetings from what was sunny and 50-degree Buffalo today. I kid you not. So it was very lovely to have what felt like a spring day in what, as you can imagine, has been a somewhat longish winter, but everything at SEM has really been amazing throughout this whole adapting to COVID, and we feel very fortunate to have an amazing faculty and head of school that have supported, our students during this time. So thank you all for being here. My name is Molly Green. I'm the Director of Institutional Advancement at SEM. I'm also class of 1998 at SEM. I don't feel I need to do too much introducing of our three speakers today, as you've all seen our invitations, but we are so thrilled to have Carrie Wick, class of 1976, moderate this discussion about COVID-19 with Perley, Dr. Perley, class of 2004, and Dr. Suzanne Bradley, class of 1973. They'll speak about their experience with the disease. Juliana, please jump in if you have any other housekeeping, but you can keep yourself on mute if you wouldn't mind. Speaker view might be easier for you to get an idea, obviously, who's speaking throughout the course of the program. And then please put any questions you have at any time for our speakers in the chat. Juliana will be moderating those, and she just indicated that as well in the chat. A good example of how you can pose any questions tonight. So thank you so much for joining us. We're thrilled to have you here. And with that, I will turn it over to Carrie Wick.
Thank you. We're very glad that you all have joined us tonight. We're excited to have this event. This is the second of the second session of new SEM speaker series that we started this year. It's an event that's organized by the Heads Council on Alumni Engagement. And we are just always looking for ways to bring SEM alums together from many different classes to just get a chance to meet each other in new ways and talk and learn about what everyone's doing across the country and across the world. So we had about 80 people sign up for tonight. People are still dribbling in, I think. But it's a nice mix of alumni. It's people are here from classes in the 1950s through the 2010s. So it's great to have everybody here. I think it's going to be a really interesting hour with our experts. As you know, our guests tonight, our speakers are Dr. Suzanne Bradley, class of 73. Sue is a physician. She's also a professor of internal medicine and an expert in infectious diseases, especially infectious diseases in older patients. And Casey is class of 04, and Casey is a civilian biodefense scientist with the U.S. Army. Her expertise is in deadly pathogens and also in development of vaccines for these pathogens, including RNA vaccines. And I think you all are familiar with the RNA vaccines that are now out in use against COVID. So I think they're going to have a lot to say, so let's get right to it. And I'd like to start with both of you, Sue and Casey, just giving a little bit more color background on what you do, where you're working, and also how you got to where you are today from 205 Bidwell Parkway. So maybe Sue, could you lead us off, please?
So yes, I was a biology major, a budding biology major at SEM, and I always thought I wanted to be a doctor, but there weren't a lot of women who were actually in the field. But I grew up wandering around Millard Fillmore Hospital, which I understand is now bulldozed, and pursued things through college, and then went to UB. Met some wonderful people there who, like maybe some of you might know Nancy Nielsen, who was interested in infectious diseases, and some other people, and said, gee, they came in, they walked in, we had tough cases. They knew right off the bat what the patient had, which had perplexed us. And I said, I want to be like those people. And I met other people along the way as I went to Ohio State and then doing my fellowship at Michigan. So that's how I ended up in infectious diseases. And I went into, did aging as well. In medicine, you need a gimmick, and I needed a job, and the geriatricians needed physicians. So I found a niche in taking care of older adults with infectious diseases that led to vaccine studies, looking at other ways to prevent infections, and the like. So it's been 35 years in Ann Arbor. I thought if I stayed one place for 5 years, that would be pretty good. It's been quite a run. I thought when we had the pandemic in Hong Kong, pandemic flu, that would be my career pandemic. I didn't realize that I was probably going to be ending my career in the midst of our worldwide pandemic called the coronavirus. So I'll pass things off to Casey from here.
Thank you.
Thank you so much. So I, like you, was a budding biology major doctor at SEM. And when I went off to college, I majored in molecular biophysics and biochemistry because I didn't want to have to choose what science I liked best. I liked all of them and I got to take all of them in that major. I also realized in college that what I really wanted to do was research science. I was much more interested in how diseases worked on a molecular level than treating patients. So after I graduated from Yale, I spent a year as a research tech at MIT doing computational biology. Went to Duke to get my PhD in molecular genetics and microbiology. And it was in grad school that I really began working in hot zones. So if you've seen the movie Outbreak, if you've seen the movie Contagion, those people in big suits, that's what I did. And it was never because I particularly wanted to wear a person-shaped bubble. It was because the questions I was really interested in answering were fundamental questions. How does this disease work? What cells are infected? How do you make a vaccine? And for diseases that are easy to work with, things you can just work with on a bench top, a lot of those questions are answered. But for things like tuberculosis or plague or hemorrhagic fever virus, because they're so hard to work with, there aren't a lot of people studying them, and you get to ask those really big, thought-provoking questions. After grad school, I went and did my postdoctoral work at the Army's Medical Research Institute of Infectious Disease, where I actually went into biosafety level 4, which is the hottest of the hot zones. We have 7 of them in this country. Spent about 3,000 hours between grad school and my postdoc handling these deadly pathogens, specializing the entire time in making vaccines and therapeutics against zoonotic infections, or diseases that jump from animals into humans. At the end of my postdoc, I took a job with Army Futures Command. I am the technical director for an innovation cell where I touch everything from human performance in biomedicine to robotics and AI. I get to supervise a great team of scientists and engineers, so I'm in a much more generalist role right now, but certainly, spent formative years of my career working with these pathogens and designing vaccines against them.
Thanks, Casey. So you've dealt with many different kinds of emerging pathogens. Is there anything in particular about SARS-CoV-2, the virus that causes COVID? Is there anything about that virus that really surprises you, or just is very different or unique that you're finding out about?
So there's 2 things about COVID that I find very interesting. The first is oftentimes when diseases jump from animals to humans, they don't make the jump all at once. So if you look at, say, Ebola in Africa, you see these isolated outbreaks that we're able to then get under control, and then it comes back. So you see it essentially, like I said, in these little spots that can come and then burn out. It's very rare for a disease to jump from animals into humans, be able to be so transmissible between humans, and spread so globally so quickly. So that's something that's fascinated me. In addition, coronaviruses in general, I find really interesting because there's some, like SARS and MERS and COVID-19, that can, have high mortality rates, that spread really easily, that make people very sick. And on the other hand, you have coronaviruses that just cause the common cold. So how do viruses that are so similar cause diseases that are so radically different? Those are the questions that always fascinated me, and COVID is just another piece of that puzzle.
Thanks.
It's interesting that you know, when the original SARS happened back in, you know, 2002, 2003 in Hong Kong, there weren't a lot of people who were interested in coronaviruses. As you said, it was a common cold. We were totally caught flat-footed. There were very few people who very quickly gained a lot of respect and grant funding because they were the few people who had bothered to study these uninteresting bugs. And to have them cause the diseases that they did in Hong Kong and then in the Middle East, and then disappear, as you said, and we really don't understand why, is really, really, really fascinating.
So as I read about what's going on with COVID, a lot of scientists are saying that it's going to be with us for a long time. It's going to become endemic. It's going to circulate around the globe and crop up over and over again in vulnerable populations. So what does this mean for us going forward in terms of how we manage it, how vaccines are developed and implemented going forward? Do you think we'll have annual vaccinations like we do for flu, and the vaccines need to be tweaked every year for the current dominant variant strains? How do you see this? I mean, not that we've gotten it behind us quite yet, but what do we expect in the coming 5, 10, 20 years with this thing?
I think one of the fascinating things when I first started reading about COVID-19 and then going back and looking at the coronaviruses, the question was, how long did immunity actually last? And I think we still don't really know that. I think I found maybe one little pediatric paper in transplants where somebody tried to actually look at duration of antibody. And I think we still don't know if it's just antibody or if there are other components of the immune system that might be helpful here. The problem is we can measure antibody easily. The other components, T cells and B cells that you might have heard about, not so easily done in outside of, the clinical labs can't do this. So still lots of questions to be answered. So the duration of the vaccine may not be that long. Perhaps it's going to be an annual basis kind of thing like influenza. I think it's hard to know. Ideally, it would be nice to have the vaccine work. We have everybody vaccinated. The virus has no place to hide out, and it slinks away and goes back to its animal vectors just to wait, reservoirs just to wait to emerge again in some other form later.
It's exactly right. And I think one of the things to remember about COVID-19 is this isn't like smallpox, where we can completely eradicate it from the globe, because smallpox has no known animal host that it can hide in. Even if we were to vaccinate all of the human population, COVID-19 can live in animals. That's how it came into humans, that spillover event, that jump. So even if we were to vaccinate the entire world, it's still going to be lurking out there. And when it goes inside an animal host, it can mutate and it can change, and it can come back in a form that maybe our vaccines don't protect against. In addition, as was said, we're not entirely sure how long protection lasts. And depending on the coronavirus you look at, you're going to get one answer for SARS, one answer for MERS, one answer for the coronaviruses that cause the common cold. And you just don't know where COVID's going to fall in that spectrum. As scientists, we're not really good at saying, I don't know, right? That's hard for us. We're scientists. We always want to find the answers. But so much about COVID is we don't know yet. We need to wait. We need to see. We need to get the data. So it's kind of hard to predict.
One of the good things is that we have vaccines now that you can, because they're molecular, and that sounds scary, but and Casey can talk to this more because she does this for a living, but you can manipulate the mRNA vaccines a lot easier than some of the old-fashioned ones, where we basically took a virus, chopped it up, inactivated it, and inject it into people and hope that we got an antibody response. You can tailor this much more precisely to what's going on in terms of the variants that are circulating. Or maybe I've made it sound too easy.
No, you haven't. I mean, so when you think of vaccines, the ones that you're all used to getting, things like MMR, diphtheria, DPT, your tetanus vaccine. There's three main types of vaccines we've had in the past, and all of them have been, we make a virus or a bacteria unable to cause you harm, even though it's alive, called attenuation. As Suzanne said, we chop it up and we inactivate it, or we give you a tiny, tiny little component mixed with some adjuvant to give you an immune response. All of those take time. And every time the pathogen changes or tweaks itself a little bit, you've got to go back to the drawing board. These molecular vaccines take a different approach. So if you think of your immune system as like a police force, and it checking out your blood vessels at all times, making sure everything looks normal. The way a cell signals everything is normal here is that it chews up some of the protein that it makes, and it puts it on its surface like Christmas lights. So your immune system can drive by, go, hey, that looks normal, and keep going. What these molecular vaccines do is they trick your cells near the injection site into making just a little bit of the surface protein of COVID-19. That protein then gets chewed up slightly, put on the top of your cell like, those Christmas lights, and the immune system recognizes, oh, hey, that's foreign. That's not supposed to be here. And it mounts an immune response. And when you get a booster, what happens is it sees it again, kind of like facial recognition software, and goes, I recognize that's foreign and that's bad, and I need to, get rid of it. So as we continue to develop these vaccines, we can tweak them a little bit for these common variants. Maybe we get a booster shot next year that's got 4 or 5 different versions of that spike protein mRNA in it that it tricks our cells into making, so our body learns to recognize it. We can turn them around in about 3 weeks once we get the sequence of these pathogens, as opposed to months to years for all of the other approaches. So these platforms, aside from just giving us a great COVID-19 vaccine that is highly effective, really have the potential to revolutionize how we make vaccines against potentially pandemic pathogens in the future, or maybe even, cancer therapeutics and other types of things where the ability to personalize medicine. They're really, really a fantastic tool, and I would expect one day in the coming decades to see them win a Nobel Prize. This is just kind of their coming out party onto the world stage.
How many of you have already been vaccinated? Can you wave your, got a few, good. Okay, Pam, Liz, I'm coming to visit shortly. If New York State will let me, or I'll come via Ohio, but in any event, so how many of you were worried that this was happening too fast?
The vaccines?
Yes. Okay. So if you think about it, so in 2002, we already started to have some idea how the virus attached to us. There was a protein called the spike protein, those little appendages that you see on the coronavirus. Those are the things that basically adhere to your cells and allow the virus to get in. So we knew that based on SARS work, and so already 18 years ago, they were starting to plan for the worst in terms of the epidemic wasn't going away, planning for vaccines. So they already knew how the virus was doing its dirty work. And were ultimately, so we already had some idea how things worked, and then MERS came along, even more impetus as we saw disease in the Middle East and Korea. Maybe we need to get going on this. So they were able to isolate that spike protein. So rather than chopping things up in crude a gamish, if you will, and with a plan of injecting it into people. And actually that's how some of the vaccines in China have been still produced that way. They actually have taken that very purified protein and they know the genetic sequence. So we're already poised to develop vaccines in a variety of different ways. In the meantime, some of these other technologies that came along, like mRNA, or there are certain viruses that are used to get spike protein into your cells, or the mRNA into your cells by various means. Those were already ongoing. So we were poised to have vaccines fairly quickly. And you'd say, okay, but trials take forever, right? Well, a typical trial, if you had the shingles vaccine, any of the shingles vaccines, it's pretty normal to have 30, 40,000 people in a trial. Not uncommon. But why does it take so long? Well, if you're waiting for somebody to have shingles, as you get older, your risk of shingles increases, but you have to wait for that disease to happen to know if the placebo versus the vaccine actually works or not. When you have a lot of disease, when you have a vaccine for COVID-19, and you have so much disease going on that the attack rates are, 16, 20% of the population, in fairly short order, you can have an answer. Did the person who get the vaccine have more symptoms than the group that got placebo or not? Did they get an antibody response or not? Did they have evidence of infection or not? Were they hospitalized? Did they die? So very quickly, you could do those trials quite rapidly. So they would have been done in the same fashion as any other trial. But we were poised to do that, and the high attack rates of the disease allowed us to get an answer pretty quickly. And for 3 vaccines, it's pretty amazing. And there's a 4th one on the way.
I also want to highlight that everybody knew the money was there. So when I was developing vaccines against hantaviruses, which are a pathogen you've never heard of because they primarily affect Asia, we were really at the mercy of our grant funding. So they would expect us to do a series of preliminary results, give them the data back, and then we have to wait to see if they gave us more money to do the next set of studies, working all our way up through clinical trials. If somebody had given us a lump sum up front, we could have gone a lot quicker, because studies that we ran sequentially, just because we didn't have the grant funding to do it, we could have run in parallel. So you also saw a lot of that with these vaccines. Everybody knew the money was there. So there was no concern about running things in parallel and moving as quickly as we could, because you had to be concerned about grant funding coming down the line.
So it looks like Katie.
I was just gonna.
Oh, go ahead.
I was just gonna see if Kate feels like her question is answered with, was it faster than you would have expected? Since I feel like you two touched on that. Kate, feel free.
I think initially I was asked so a year ago, we started having our incident command at the hospital. They said, how quickly do you think we'll have a vaccine? And I said, probably a year and a half. So this is faster than I thought. And to have 3 vaccines with the kind of efficacy that we have is actually pretty spectacular. Remember, the benchmark for successful vaccine was 50% protection, and we have 95%, 74%, 84% reduction in hospitalization, 100% protection from death. That's pretty spectacular.
I agree. This is faster than I thought, because I didn't think the first one would work. The fact that the first 2 vaccines that made it through clinical trials had such spectacular success rates, I wouldn't have guessed happened. I would have guessed the first 1 or 2, maybe we're at the 40, 50% range, like maybe just sort of creeping into the realm that we considered good. But yeah, Moderna and Pfizer were absolutely spectacular with the results they produced.
So the other question was about, there's this concern about the vaccine affecting fertility, particularly related to the placenta. They are this spike protein, and I've forgotten the name of the protein in the placenta, but they both begin with S. They're 2 completely different molecules. They have no relationship to each other. It's something that that has come up on the internet, but there's no basis in reality for that concern.
I want to echo that. I also want to say, especially these mRNA vaccines, the mRNA is not very stable. It does not stay in your body very long. If it did, we'd have been able to cure a lot of other diseases that have genetic components, because it would have solved some problems for us that we're having with gene therapy. So the mRNA just can't stick around your body long enough to do any long-term damage like that.
Let's see, there's a question about Pfizer and Moderna, or mRNA, versus J&J, which uses a virus that's been basically genetically altered to help deliver the genetic material for the spike protein into cells. I think one comment about J&J, the adenovirus has been used in a lot of other vaccines. Ebola vaccine, Casey can talk to this. I'm a little biased because I got Pfizer, but I didn't have a choice, and I would suggest, like Dr. Fauci would say, whichever one shows up first, go get it. There are advantages. I mean, J&J can be stored in a standard refrigerator, which will help delivery tremendously in parts of the country. At our hospital, we're literally taking coolers with dry ice and taking them out to outreach clinics and places where when people can't get to the hospital, where we have our -80 freezers. So I can't really, in terms of the delivery systems, as a non-molecular, well, I tinkered with molecular biology, but I am not a molecular biologist. I don't have a strong opinion. I think the bottom line is, when you're looking at, the bottom line is, we don't want people in hospitals and we don't want people to die. Those are and all three of them have the same profile in terms of preventing no deaths in the treatment arms, and 80% plus prevention in hospitalization, and that's the key for all three of them. You can argue about 95% preventing symptoms. You can argue about versus 74%. But the bottom line is, we're really talking about death and hospitalization.
Absolutely agree. You hit on the head everything I was going to say, which is get whatever one you can. And also, if it's your turn to get vaccinated, don't feel like, I feel like there's other people who are more deserving than me. Maybe I should keep waiting. At this point, if it is your turn to be vaccinated, what we need most is herd immunity. So go out and get that shot, and get whatever one is available to you. I also got Pfizer. I'm a little biased towards Pfizer and Moderna just because they're mRNA vaccines, and I worked on mRNA vaccines. So I like seeing the technology platform that I used do so well, but I would absolutely get Johnson & Johnson if that's what was, available to me. I will also say, when people ask if I think it's safe, I encouraged my mother to participate in the Pfizer clinical trial because it meant there was a 50% chance she was going to be vaccinated in November. All right, 50-50 chance she's in the placebo group or the vaccine group. And I felt safe enough in the technology and in that vaccine to say to my mother, go be in this clinical trial, right? There's no higher level of safety I can give it anecdotally. Unfortunately, she was in the placebo group. My gambit failed. But I'm biased towards Pfizer because my mom was in that trial, and I'm really, really happy to see that it worked.
Let's see. I'm not sure I can read all the questions are rolling in.
I'm happy to help with questions.
Okay. Yeah.
So
go ahead.
Don't feel inundated by the questions. I'm happy to interject.
Okay.
If there's anything else you want to touch on, Carrie, if you have any questions, please feel free, and then I can go back and forth to the chat for you so you don't feel overwhelmed.
Well, I think there was a question about whether this could have been developed in a lab, and again, I think I'd have to defer to Casey on this. My understanding, and I have to confess, I didn't read the Nature and Science articles in huge detail. I tend to read the bottom line. But information has just been rolling in so quickly that you can get buried in the details. My understanding is there were no evidence in the isolates we had of when you take a piece of DNA or RNA, depending on the kind of virus, and you insert it into a virus, usually there's some telltale signatures, what they call insertion sequences and things like that. And my understanding, at least initially, was there was no inkling of any sort of manipulation. And that doesn't come out in the media much, and I don't understand why.
So you're absolutely right. When people talk about, did this come from a lab in Wuhan, China? There's realistically two different scenarios they're referring to. One is, could there have been an accidental release of a natural isolate that they had in their facilities? And the other is, could it have been like genetically engineered bioweapon that got out? So speaking to how likely is it that something in one of these high-level facilities is accidentally released? Incredibly unlikely. Just as an example, in the facilities that I worked in, our air was triple HEPA filtered before it left the facility. All of our waste was treated with bleach and then heat treated. Anything that came out was sterilized. I had to come out in a 7-minute bleach shower and then take a personal shower and leave all my scrubs in between those 2 showers. So there's no way that real accidental releases can happen on this large of a scale. And something that is intentional, like somebody carried a vial out of the lab, also just incredibly highly unlikely. As Suzanne said, when you take a look at the sequence, there's 2 ways we know that this isn't like a man-made genetically altered thing. One is that there are known of those telltale like insertion sequences where we can see that a scientist has put something into a virus. In addition, while we know this virus is related to SARS-CoV and MERS-CoV and the other coronaviruses, it differs in some key respects. And the ways that it differs is not the ways we thought it would differ. And there's no way, like computational biology wise, we would have predicted the way that this virus looks. This is Mother Nature evolving herself. This is not a scientist coming up with an idea, because there's no way a scientist would have had this idea, because this is like a very roundabout way of getting at something. So, I mean, as definitively as one can say without having monitored the Wuhan lab 24 hours a day for years, this really came from Mother Nature. This isn't a release, accidental or otherwise.
And we had 2 other natural experiments. We had SARS-1 and we had MERS. It happens.
Thank you. The next question's from a 1973 grad, Lori Littlewood Coleman. She wants to know if it's safe to travel after you have had the vaccine for 2 weeks.
So I think the CDC will probably change its recommendations. I think if you look at the recommendations, I think they're confusing enough that they're attempting to ask you not to travel in the near future. For example, if you want to go overseas, when you come back with 3 days of leaving that site, you need to be tested. And then when you come back, ideally you're supposed to be tested, either quarantined for 14 days, which seems ridiculous for most of us that don't have that much vacation. And then you get tested like at day 3 or 5 after you get back, and then, but you're still off for like 7 days. It's very confusing. I think, I'd love to drive to Florida, Lori, and I know you do too. I've thought about flying, but I might drive if my sister gets vaccinated. The folks have been, but she hasn't been yet. I'll probably wait.
I just really want to be able to see my kids. I haven't seen them, so.
So when are they going to get vaccinated? Do you have any idea?
My daughter got it today, which is great.
Okay.
She's in Denver.
Okay, that's a little bit of a trip.
Right.
I think we're getting more and more information that people who have been vaccinated don't have asymptomatic shedding, so that's reassuring. So give her a chance to get her immunity up, and then maybe by that time we'll have more information.
Right.
And then you'll have to decide if you're going to hike it or if you're going to fly.
All right. Thanks.
More information is the key, right? I said early on, scientists hate saying we don't know, but we have to say we don't know a lot. And I expect as we get more data, we'll see shifts in the CDC recommendations based on what we know.
It's constantly been shifting. I'm looking forward to the day that I don't have to go to the CDC website every morning to see what they've been up to.
I'm sure Helen Marlette feels the same.
And how they have changed the recommendations and all of that. At least we've had some happier recommendations in the last 48 hours. So
Sue, me too, but I have to say both Sue and Casey, I was fortunate enough to be able to pick up the phone and say, what the heck? You know, I'm getting mixed, and Casey, we were on a Zoom call, and I think I was on the golf course when Sue called me back once. And it was like, I am taking this call because I am trying to figure out what is the right thing to do. And thank goodness we had the connection of SEM women. And this is fascinating. I'll bow out now, but I just wanted to thank you two for helping SEM during this time.
right, to do it. Something I know how to do, Helen. Not everything. In any event, let's see. There's a question about the VA.
Yeah, Sue Henderson, 87.
Susan, yep. And Susan, I haven't had a chance to email you back. So I think one thing that's been really great about the VA has been you have a bunch of people who have military experience who, if you give them, and we get a lot of bad rap, but if you give them a mission, whether it's the guys who clean the floors or the people that order the equipment, or you want to roll out a vaccine, we've delivered some 26,000 vaccines since the end of December. We have contacted all of our primary care patients. That's some 65,000 people. That doesn't mean they've necessarily received our calls, but we vaccinated probably 55% of those individuals. About a third have refused, and we're still working to get a hold of the others. So I've been very impressed. A group that I couldn't get to take an influenza vaccine in the previous 11 years that I've been doing infection control have really stepped up and embraced this process. We have drive-through. We bought 2 modules where people can drive up. There's a space where they can sit for 15 minutes. We have drive-through testing. People have been very creative and very receptive. So I hope it's been the same in Atlanta and in DC as well.
Yeah, thanks for that answer. My husband is a veteran, and we got vaccinated through the state of Maryland, but we hadn't heard about the VA in DC. But I have a friend who works there as a lawyer, and she said that the process has been just as impressive. The rollout has been really good. And another thing that I think is that needs to be publicized more. I mean, the VA has a lot of problems, but they're also, it's a great system, and when you want to get things done, you can get them done. And so I'm looking forward to reading more publications about, the success of the VA.
So one thing we did at my place early on when we had our outbreak, our director was very supportive. We had the ability to test serology very early on to see what proportion of our healthcare workers had been exposed to the vaccine. We wanted to get an idea, had been exposed to the virus rather. We wanted to get an idea how good were our infection control methods, how good was our PPE. And we didn't have any lack of PPE, unlike a lot of hospitals. We were very, very fortunate. And the director, we made it happen. People wanted to know, probably, I would say 2/3 of our employees signed up. They were enthusiastic. And the interesting thing was that our rates of COVID exposure were less or the same as in the community. So there wasn't an increased risk of getting COVID by working in our hospital that you might think. And that was very reassuring to the staff. And we were able to publish that early on, which was nice, but it took the support of our leadership, and that was really great.
Carrie, or Casey, did you want to add anything to that?
The military does logistics really well. So it doesn't surprise me that the VA is doing well. And I think that's also why you saw the former Army Materiel Command commander, who is in charge of like all logistics for the Army, take over Operation Warp Speed. Military and logistics, they do go hand in hand. So we, as a civilian in the DoD, in the DoD, we were glad to do our part as well. And I know our soldiers that have worked on Warp Speed are very proud of the work that they've done.
One thing I'm hoping is that we learn from this. When we talked about pandemic influenza, you remember influenza, years ago, we were preparing for that. And there was a lot of discussion, not only about how do you get drug companies to make vaccines if nobody buys them. Well, if the government pays for it, then you've got to buy in. So we've learned that. The other was the rollout. We have 50 states doing 50 different things. Ontario, I believe they still have a provincial system for rolling out influenza vaccine. So they would be prepared to do something on this scale. That's the hard part. Putting it into bottles and shipping it is the easy part. It's getting it into arms that's the tough thing. So I was not surprised that we were having trouble getting people vaccinated nationwide. That it's just about infrastructure and having the will to maintain it.
Historically, the last major vaccination campaign the US was a part of was smallpox, polio about the same time. I remember making a comment to my parents at the beginning of the pandemic, you know who we need? We need to bring D.A. Henderson out of retirement. He was the gentleman that had ran part of the global smallpox vaccination campaign. And I looked up and I realized the reason we hadn't brought him out of retirement is unfortunately he passed away. And that a lot of the scientific knowledge in this country about how to run mass vaccination campaigns like this had passed because it had been so long since we'd done it. So we're educating a generation, multiple generations of clinicians and public health experts and state health officials who now know how to do this. So in the future, if there is pandemic influenza or if there's something else that comes up, we've gained this expertise that we really didn't have at the beginning of this because it had just been so long.
Let's see, there's something about funding. I'll do my best. So Pfizer didn't take any money from the government, sort of like some car companies and others. So the question is, so they get money to make the vaccine, and I presume that they're going to make money off of the vaccine, although it's not any cost. You guys should not be paying for it. There may be costs associated with maybe your provider. If you get J&J, for example, and your provider has to store it and administer it, there may be some costs that way. But yeah, I'm not, as Helen knows, I'm not much of a financier. I don't know if you've got any more insight on this, Casey.
Some vaccine companies took federal money, some did not. That was a choice the companies made, but I think all of them knew at the end of the day there was going to be a market for it if they had a successful vaccine. So beyond that, I don't have any insight.
Yeah, but Merck did not succeed. AstraZeneca is having some issues. I don't know, Carrie, if you have any
Carrie might have some
comments
insight.
about.
Yeah, it's interesting because traditionally, pharmaceutical companies have not made money off vaccines, and they really don't want to make vaccines because there's no market for it. So the government has propped up vaccines for a lot of diseases for a long time. And then I think pretty much in the 70s, there were a lot of lawsuits about injuries from vaccines, and pharmaceutical companies were saying, you know, we can't deal with this. We're out of this business. And so again, the government had to create a system to protect the pharmaceutical companies, to basically lure them back into making vaccines. So that system is still in place. There's a whole adverse event reporting system and compensation for people who have been injured. That's a very separate system from the regular tort law for injuries. And I think what's interesting, especially with the RNA vaccines, is I don't think there's any information out now about who's making money or not making money off these vaccines. But I think what's really interesting for the RNA vaccines is that this is beautiful proof of concept. These RNA vaccines, this is the first time an RNA vaccine has been used in humans. And as Casey was explaining, it's kind of it's almost like a cut and paste system molecularly, where you can take this technology and use it for different parts of the SARS-CoV-2 or different viruses. And now that this has been just beautifully demonstrated on a large scale in humans, that I think we're going to see a lot more vaccines based on RNA RNA technology. I think also with regard to developing vaccines for the next pandemic, whatever it may be, this was very interesting that the government just went full force and did such huge amounts of funding so fast for vaccine development. But unfortunately, it just seems like there's never any action until the next pandemic starts. And I think that Casey might be able to comment, but I think we're going to have another flu pandemic at some point, and we could have other coronavirus pandemics. It's not to be a fear monger, but the more that human populations interface with wild animals and then encroach on formerly wild turf, there's more opportunity for jumping from animals to humans. And I wish that there would be just a more proactive program to start developing technology platforms and vaccines for a lot of these viruses. We have a pretty good guess that they could become pandemics, especially for flu, because we've seen that happen 3 times in the last century, and in 2009 most recently. So that's kind of my little soapbox, that we have the technology, we kind of know it's going to happen, and there's the system for funding vaccines for diseases that aren't yet a problem but probably will be. It's just not there. There's no commercial market, and the government just is unable to, I don't know, they have other priorities, and they just won't put a program in place to defending against potential threats. We tend to wait till it is a threat or it's real.
It's like funding public health or infection control.
Yeah.
I can tell you, running an infection control program, I put in a request for a second infection control practitioner 3 years ago. It took an international pandemic to finally get it approved. So in any event, shout out to Ma Ellis, Pammy, give her my love, and make sure she gets vaccinated. Let's see, what else do we have?
The next question is
ahead.
about longevity of the symptoms, and if the vaccine.
The long haulers.
Yeah, the long haulers, yes.
Yeah, I think this is one of those we don't know there's probably some intense immune response happening with the long haulers that we don't completely understand. I just don't think we have enough information at this point to even comment.
I would agree. I mean, once again, we don't know. We need more data. I will say there are other RNA vaccine or RNA viruses that can cause problems months and years down the line. Measles is one of them. So it's not without precedent that this can happen. But exactly what it's going to look like with COVID, how do we treat it, what proportion, all of those questions, which is what everybody wants to know. You just have to wait for the data, unfortunately.
Children under the age of 16. So, you know, one of the vaccines is approved for over age 18. The other, I think Moderna is 16, if I'm not sure. I think that's correct. I know they're doing shorter, smaller trials. I think they're talking about 2022, though, to be honest. I don't know. I don't know if you have any inside track on that.
I'm hearing late 2021 or early 2022 as well. So basically the same time frame. And I just want to emphasize that when we do clinical trials in this country, medical ethics is that you test essentially on healthy adults first, and then you move into other age groups, right? So that's why there were no deliberate trials in pregnant or breastfeeding women at first. It's why children weren't included at first. So now that the vaccine has been proven safe in the general population, now you see those clinical trials that are looking at younger children, that are maybe looking at other classes of individuals as well. So there's, I know I've heard comments about why weren't children included at first, etc. That's why it's just standard medical ethics practice in this country. And unfortunately, like everything else, it takes time.
Glad to hear the Buffalo VA is doing well. I spent a few hours there as well. Let's see. Yeah, Merck. So there are some companies have focused on monoclonal antibodies, and we are giving infusions in the emergency room to people who have had COVID for up to 10. You know, if you've been tested within 10 days, you can get an infusion of some various monoclonal antibodies. There are multiple preparations. I wouldn't be surprised if Merck has one. I've been so focused on infection control. Until I go on service in June, I'm just going to let things sort of settle in terms of treatment. But yes, the monoclonal antibody issue is a huge one for inpatients and outpatients as well as a treatment. In addition to steroids, remdesivir, I'm sure you've heard about, there are a number of things that we use. Treating viruses is very difficult because they're intracellular pathogens. So if you kill the virus, you could kill the patient as well. So it kind of limits what we can give. We certainly have to be careful with the kinds of drugs that we give.
We just have.
I just want to say one thing on monoclonals. So monoclonal antibodies, if you think back pre-antibiotic era, or you think to Ebola where we don't have drugs to treat it, one of the things you hear a lot about is convalescent plasma. So somebody that's been sick and survived donates their blood. And that's because we really want the antibodies that are in that blood. Monoclonal antibodies are just a much more effective and targeted preparation of the same idea type of therapy. And the way they work is that they bind to the spike protein of the virus and they stop it from infecting your cells. So that's how that treatment works. Like I said, it's just a variation on something that's been around, since the 1940s when we were using horse serum or serum from individuals who'd recovered. So it's a great thing we often go to early on in epidemics is to try to find those antibody cocktails. I did a lot of work on that for hantavirus in my postdoc. It's another therapy that I firmly believe in. Charlotte,
long time. Hope you're doing well. Appreciate you being here. Susan, yes, in fact, so when we treat patients with COVID in the hospital, they're very sick. They have all sorts of devices, ventilators, IVs, all sorts of things. So secondary bacterial infections are a major issue. I run an infection control journal called infection control and hospital epidemiology, and I've just accepted a paper from the CDC that talks about the increase in bacterial line infections in COVID patients. It's a major issue across the country. And every hospital, including mine, is held to account when we see an increase in these infections. It's certainly a change.
I was just going to say, we usually wrap up in about an hour, which is just a few minutes away. So if Carrie, Casey, or Sue, if you guys have anything else to add or any last minute questions any of the attendees would like to address before we end the night.
One thing that I wonder if Casey might want to touch on, and Sue as well, is of how this virus is mutating so fast. What's going on? Is this unusual, the way it's mutating and developing these strains that are potentially more transmissible? And is it possible that the monoclonal antibodies and or vaccines could actually be pushing the mutations in one direction or another?
So that's a great question. So I'm going to ask you all to remember back to your SEM high school biology classes, right? DNA, it's the recipe for you, makes RNA, makes protein, right? And one of the reasons we have that RNA step in between DNA and protein is so we don't damage the DNA, which is the genetic material that encodes everything about you. COVID is an RNA virus, which means the RNA serves both as its genetic material and as the basis to making the proteins. When RNA viruses replicate, they're about 100 times more likely to mutate than DNA, which means the more that COVID-19 replicates, the more likely it is to mutate. When COVID mutates, it's going to have one of three types of mutations. A mutation that is deleterious for the virus, meaning it did the virus actual harm. Those strains don't survive. You're going to have some neutral mutations, no effect, good or bad, right? And those will start to creep into the mainstream. But the ones that you'll really see and the ones you'll hear about are the ones that give COVID an advantage. And the most likely advantage is one that makes it spread easier, make it be more transmissible, because the more easily a virus is spread, the easier it spreads and the larger it spreads, right? So the fact that we're seeing these mutations is absolutely not surprising. The fact we're seeing these mutations lead to increased transmissibility, also not surprising. Now, the biggest concern about this is it going to cause the vaccine or these therapeutics to become less effective? That's always a possibility, especially if it affects the spike protein, which is what our vaccines and our therapeutics typically target. It's most likely not going to be an all or none phenomenon. You're not going to go from your vaccine being 100% effective to your vaccine being 0% effective. But you might start to see some decreases. You also might start to see cases where, the vaccine could protect against 100 times the infectious dose of the wild type strain, but only 80 times or 60 times, the infectious dose of these new strains, which is going to lead to the vaccines becoming slightly less effective and maybe us needing a booster shot that contains some of these variants in it. So I hope that answers the question. If anybody has further questions, I'm happy to follow up.
Again, I think we're hopeful that we're getting the vaccines rolled out, that the vaccines are currently highly effective, and even if some of these strains are less effective, less active against the antibodies generated by our vaccines, that would still be enough to prevent hospitalization, death, and symptomatic infection. But there's a race here. The longer we wait to attain herd immunity, the more the possibility increases. So it's a matter of time, and we'll have to see about the monoclonal antibodies as well. People are certainly looking at that, and there are newer formulations coming out all the time as well. So we'll see. But yeah, I wish I could say that on August 5th, everything will be done. 100% of people will be vaccinated. It's probably not going to be as dramatic as that.
I just want to piggyback on one thing you said. The closer we get to herd immunity, the more people that are vaccinated, the less of a chance this virus has to replicate, which means the less of a chance you get to see these variants. So the best way to prevent the variants is herd immunity and to get the vaccines that we have now. So.
Hi, I was just curious your thoughts on the flu being dramatically down this year, if indeed it is, or is it being hijacked or masked by COVID?
So Michigan has a very active flu laboratory. Arnold Monto is a guy you may have seen on the news. They're constantly reporting on flu. The VA tracks it as well. We are just not seeing it. I think we've seen one case of flu in our hospital. And I think it's real. We are using the tests that we do for COVID. There are certain ones, in fact, both of them that we use in the hospital detect flu viruses as well as COVID. We are just not seeing it. So whether it's, people are masking up, don't know. It doesn't mean it's gone, it's just hiding. But I don't know why.
Great. Well, thank you everyone so much for coming tonight. This was fascinating to me as a self-proclaimed, not necessarily science person. Thank you for putting this, what can be really complicated information, in terms that we can all understand. And then just particularly amazing that all three of you are such accomplished and intelligent STEM graduates, as all of the STEM graduates here tonight are, but really awesome to hear from all of you. Thank you so much. We'll be sharing this recording with everyone. So if you wanted to look back on something, you can look for that in your email. And as Helen said in the chat, we're really looking forward to, when we can travel safely, welcoming everyone back to STEM, so we can all be together again in person. But for now, these Zoom programs have really been great. So thank you again to Carrie, Casey, and Sue. We really appreciate your time and expertise, and we look forward to seeing everyone at our next program. Thank you so much.
Thank you, Sam.
Thank you.
Thanks, everyone.
Thank you.
Thank you. Good to see you all. Stay safe. Liz, Jan, everybody. Good to see you again.
Thanks.